Well, hello everybody. My name is Ross Macdonald. I'm the Chief Executive Officer of Cynata Therapeutics, and I'm looking forward to speaking with you today and to giving you a presentation on Cynata Therapeutics and the activities that the company is involved in. First, let me draw your attention to our safe harbour statement here, and to ensure that you do check our filings on our website and on the ASX, Australian Stock Exchange site as well, before you make any investment decisions or any other decisions in relation to the company. Let's jump right in then and give a brief overview on our activities. We are a clinical stage biotech company developing a proprietary platform technology for the manufacture of mesenchymal stem cells, or mesenchymal stromal cells, or MSCs for short. That platform technology we call Cymerus, and we're developing products arising from the use of that platform to treat a range of serious diseases. The technology originated from the University of Wisconsin, which is very much the home of stem cells. Many of the early discoveries around embryonic stem cells and the biology of stem cells were made at the University of Wisconsin, and indeed some of the inventors on our patents are the leaders in the field worldwide. Our first product has been successfully trialled in graft versus host disease, CYP001, and has now been licensed to Fujifilm for that indication worldwide. We expect in the course of the company's near-term future to engage in multiple further license transactions, and it will inform the market of those at the appropriate time. The Cymerus technology very simply addresses the manufacturing challenge associated with tissue-derived manufacturing strategies. As you're aware, most MSC products are derived from either bone marrow or adipose tissue, and in our view, that represents a major shortcoming or challenge for their ultimate commercial application. The company is listed on the Australian Stock Exchange. Our market capitalisation is around 70 million at the time of recording this presentation. We have good cash balance to allow the company to undertake its activities over the coming year and a half or so, and so well placed financially. Fidelity International is our largest shareholder with just under 10%, and our strategic partner Fujifilm just under 7%, and insiders just under 6%. Turning now to our portfolio here, we have a rich portfolio of phase 2 and phase 3 assets. GVHD is being developed through our partnership with Fujifilm. Osteoarthritis, a phase 3 program, which will get underway in Australia this half, we anticipate. The COVID-19 trial that's enrolling at the moment, a trial in CLI that has been approved by the UK regulatory agency, but which, given the pandemic situation, is currently on hold, and a very rich preclinical database in indications such as coronary artery disease, diabetic ulcers, heart attack, and others. I'll talk more about some of these in a moment. But let's turn now to our technology and why we differ with the Cymerus platform from the conventional approach to manufacturing MSCs. And that conventional approach is illustrated schematically here in this slide in the top panel, where, as many of you will be aware, MSCs are derived from a donor material such as bone marrow, which requires, in the case of bone marrow, a bone marrow aspirate and multiple donors. The isolated material is then expanded in culture multiple times, and then ultimately the finished product is infused into the patient as indicated there on the right. This differs dramatically from the approach that Cynata takes with the Cymerus technology, which involves the utilization of iPSCs as a starting material. And of course, that requires a sample from one donor, one time, to generate the iPSC line, from which we can differentiate that cell line into MSCs, the finished product. But it's the expansion potential or the expansion capability of iPSCs which render them most useful for cell therapy applications. iPSCs, having many of the properties of embryonic stem cells, including an ability for essentially infinite self-renewal, provide a very powerful tool for the generation of differentiated tissue, such as, in our case, mesenchymal stem cells. So this represents an effectively limitless expansion capability, requiring in turn minimal expansion at the MSC level. So ultimately, this results in a highly consistent product, effective manufacturing process in quantity and quality, and then ultimately a potent product, which in the case of GVHD has shown to be highly efficient, which, as I've indicated here, requires only 2 infusions to achieve a very good therapeutic outcome, compared with at least 8 infusions of bone marrow-derived MSCs. These characteristics of the iPSC-driven strategy have come into stark focus recently with the FDA's recent assessment of a BLA for a bone marrow-derived MSC product. And it's now become very clear that the FDA is deeply concerned about the functional heterogeneity that's been observed between MSC batches derived from different donors. And this is a point that the FDA and other regulators have been making for a long time, but now it's become very much a more formal issue with the FDA's documents in relation to this BLA. And that is that if a manufacturing strategy that relies upon multiple different donors is flawed from the outset, because every donor is different. And so therefore MSCs isolated from those donors are also going to be different, unless the manufacturer can somehow prove that they're identical. And this has not been to the satisfaction of the FDA so far. However, using the Cymerus process, we derive product from a single donor one time. And that in itself addresses the product consistency challenge associated with multiple donors. It also provides an essentially limitless expansion capacity before differentiating into the MSCs, meaning that the MSC itself doesn't require multiple rounds of population doublings, therefore maintaining potency. And ultimately, this guidance from the FDA, I think, has been very, very helpful for the industry, because it now brings into focus this area of concern of the regulator, and allows companies such as Cynata to more effectively, I suppose, address those concerns, not only with our manufacturing process, but with determining the critical quality attributes and various other specifications relating to the product. So turning now to clinical trials and whether or not MSCs manufactured using the Cymerus process are effective. And we have completed a phase 1 clinical trial in acute steroid-resistant graft versus host disease with very encouraging results. On the left-hand side of the slide, you'll see the data at the completion of the primary evaluation period at day 100. In that, all endpoints in the clinical study were achieved. A complete response, that is complete resolution of symptoms in more than half of the patients, an overall response in nearly all of them, and likewise nearly all of them were still alive at that day 100 point. And this is, it's very important to point out that in acute steroid-resistant GVHD, the outlook for these patients is typically very, very poor. So this is a great result, not only for the company, but of course also for patients and their carers and families. The response rates were higher than what we would expect to see and would be required for a phase 3. So we believe that if these data are repeated in further studies, which are now the subject of our partnership with Fujifilm, then the product should find a fairly clean pathway ultimately to market authorization. On the right-hand side of the panel, you'll see more recent data, which represents 2-year follow-up following completion of the dosing period of these studies. And in these patients at 2 years, we saw an overall survival rate of 60%, which compares very favorably with the survival rate with standard of care, which is less than 20%, and the survival rate at different time points using other MSC therapeutic products, around 17 to 40%. So whilst it's difficult to do an apples-to-apples comparison, these data really are very, very exciting and point to Cynata having a very effective product in GVHD. Importantly, there were no treatment-related AEs or serious safety concerns identified, which is very much the pattern with MSC-based therapeutics. Turning now to COVID-19, which of course needs no introduction under these current circumstances, and the increasing global attention that's being paid to potential therapeutics, particularly for patients who are severely impacted by the disease, and the potential role that MSCs may have in those patients. We know, of course, that COVID is a respiratory virus, or SARS-CoV-2 is a respiratory virus, and which in patients that have severe complications, primarily affects the lungs. And the sequelae of those patients typically is manifested in acute respiratory distress, sepsis, and cytokine release syndrome, all of which Cynata has compelling preclinical data in models of those diseases, most of which is now in the public domain and published. We've leveraged this preclinical data to develop a clinical trial protocol in COVID-19 patients with severe complications, which we call the MEND trial, and that is now open for recruitment. But interestingly, the three target diseases here, ARDS, sepsis, and CRS, represent very interesting commercial opportunities in their own right, with applications that go well beyond the current COVID-19 pandemic. The trial will be in 24 adult patients admitted to ICUs with compromised lung function. And of course, the rationale for selection of this indication is based on our strong preclinical database and an interesting set of early clinical data which suggests that MSCs may have some promising effects in these patients. It will be a randomized clinical trial, open label, conducted in New South Wales in Australia. The primary endpoints will be change in the oxygenation ratio, and of course we'll be looking at safety and tolerability. As I mentioned, ethics approval has been obtained and patient recruitment is now open. And we will assess the opportunities to expand the trial also into other jurisdictions as we progress. A further trial that the company will soon commence is a phase 3 program in osteoarthritis. And this disease also needs no introduction. It's a major unmet medical need. And this trial will be designed to assess the effect of our MSC product in clinical outcomes and knee joint structures of patients with OA of the knee. It will be a placebo-controlled trial in 448 patients, so one of the largest clinical studies undertaken using MSCs. We now have ethics approval and the study is ready to go, subject to the moratorium on clinical trial enrolment being lifted by the relevant authorities here in Australia. From a commercial point of view, Cynata sees a number of different opportunities to build shareholder value. Of course, the conventional approach is to continue to generate data, and data drives value, as we all know. So building the asset value of our platform with the generation of additional clinical data. But we can also partner our technology, and finally, we can also engage in strategic discussions. And in fact, last year, Cynata was the subject of a takeover bid that we decided not to proceed with at $2 per share. So a substantial increase in the share value compared to today's price. Looking now at a partnership strategy, which really drives the company's business strategy, and that is exemplified by the deal that we have done with Fujifilm, our global licensee for GVHD. All development costs will be met by that partner. They've already contributed significant sums to the company, and a further around about $70 million plus in upfront fees, milestones and royalties will be payable as the product progresses. Importantly, this represents a major endorsement by Big Pharma and builds ongoing relationships for further commercial agreements. So moving towards my closing remarks here, our portfolio is rich with broad application across a range of diseases. A phase 3 that's ready to commence in osteoarthritis, and one of the largest clinical studies using MSCs ever undertaken, which will finally address the question that I think many of us have, and that is, do they really work in orthopedic applications and in particular in OA? A phase 2 will get underway, we expect later this year, through our partnership with Fujifilm in graft versus host disease. phase 2 in CLI is currently on hold. And in the case of COVID-19, that trial is recruiting. But we also have a broad portfolio of preclinical data in a wide range of clinical targets as listed here, which not only validate the approach of an iPSC-driven manufacturing strategy, but also provides opportunities for further partnering. Valuations are an interesting topic, and it's very clear here that Cynata has got an enormous upside potential, particularly when compared with some of our peer companies. BlueRock and Fate Therapeutics, both developing iPSC-directed cell therapies. In the case of BlueRock in Parkinson's, and in the case of Fate, a number of cancer targets in immunotherapy-based approaches with NKs and CAR-Ts. Those companies have valuations, in the case of BlueRock, which was acquired by Bayer over at $1 billion valuation, and Fate at the time of giving this presentation with a market cap of around $2.8 billion US. So look at Cynata here at around $70 million US. Even one of our own countrymen, Mesoblast, which admittedly is further advanced, but which is developing bone marrow derived MSCs, has a much higher market cap. So we think with the next generation technology, the leading company in the world with iPSC-driven manufacturing strategies for cell therapies should be much higher valuation than we are today. So finally, let me wrap up with a summary here. Cynata is developing the world's leading iPSC-driven manufacturing strategy for therapeutic MSCs, a solid and now proven technology platform. We've been successful in our license-driven business strategy with our partnership with Fujifilm, and also, of course, in attracting the attention of a large pharmaceutical company that bid for Cynata last year. Successful outcome in our clinical studies so far in graft versus host disease, and a rich pipeline moving forward in a number of different indications, including COVID-19. Ultimately, as MSCs prove themselves to be effective therapeutics, Cynata's position in the regenerative medicine business will continue to grow, and we are very, very well placed with a second generation manufacturing strategy for the production of therapeutic mesenchymal stem cells. Thank you very much for your attention, and I'd like to conclude at this time and look forward to seeing you again in person at next year's MESA meeting. Thank you very much.